Hello friends, to the previous slides we have seen about the blood functions, form elements of the blood and WBC, its types and its functions. So in this presentation we will see the actual blood clotting mechanism. Actually blood clotting is a very protective mechanism. It is responsible for prevention of an blood loss and it can be done with the help of the very basic mechanisms like vascular spasm, platelet plug formation, as well as a common pathway of an blood clotting. I am repeating that one. Blood clotting is an protective mechanism for the prevention of the blood loss. So it can be protected, the blood loss can be protected with the help of the three basic mechanisms. We can call it as an vascular spasm where the blood vessels are getting contracted to stop the blood loss. Second, platelet plug aggregation where the platelets can come at the point of the injury and form an plug. And third one is an activation of clotting factors, and that is responsible for formation of an clot in the blood for the further prevention of the blood loss. So platelets or thrombocytes are responsible for formation of an platelet plug formation. There are the most numerous of all blood cells, and these are having in numbers 1.5 to 4 lakhs platelets per microliter of an blood. Shape is disc shape and diameter is about 2 to 4 micrometer. Normal lifespan is about 5 to 9 days. Platelets have many vesicles but no nucleus. These are non-nucleated cell containing granules in the cytoplasm. It plays a major role in the hemostasis. The prevent blood loss from damaged blood vessels by forming a platelet plug which inhibits bleeding. Also, they release certain chemical mediators which promote vascular spasm and blood clotting. Platelet contain actin myosin molecules which help platelet contraction. They also contain endoplasmic reticulum, Golgi apparatus and mitochondria. The cell membrane of platelets contain a coat of glycoprotein on its surface that avoid its adherence to normal endothelium but make it adhere to injured areas. What is meant by process of hemopoiesis? Hemopoiesis is nothing but an formation of an red blood cells. Thereby you can say that it is responsible for formation of an blood. The process by which the form elements of the blood developed is called as hemopoiesis. Red bone marrow is highly vascularized connective tissue. Approximately 0.05 to 0.1 percentage of red bone marrow cells are derived from mesenchyme and are called pluripotent stem cells or hemocytoblast. In order to form blood cells, pluripotent stem cells produce two types of stem cells which have the capacity to develop into several types of the cells. These stem cells are called myeloid stem cells and lymphoid stem cells. Myeloid stem cells begin their development in the red bone marrow and give rise to red blood cells, platelets, monocytes, neutrophils, eosinophils and basophils. Lymphoid stem cells begin their development in the red bone marrow and migrate to lymphatic tissue where they differentiate into lymphocytes, like in types of T cells, B cells and natural killer cells. During hematopoiesis, some myeloid stem cells differentiate into progenitor cells which are committed to forming specific blood cell types. So progenitor cells are nothing but which are committed to form a specific blood cell types. So other myeloid and lymphoid stem cells differentiate into precursor cells. Precursor cells are committed cells that further develop into specialized blood cell types but cannot reproduce themselves. Certain progenitor cells are called colony forming units. We can call it as a CFU. Okay, CFUE leads to the production of erythrocytes, that is red blood cells. CFU MEG leads to megakaryocytes. So MEG stands for megakaryocytes, which form the platelets, and CFUGM leads to granulocytes, which forms the granulocytes, neutrophils, eosinophils, and basophils, and monocytes. So granulocytes containing neutrophil, eosinophil, basophils, as well as the monocytes. AA progenitor cells advance in differentiation. They become precursor cells, often referred to as blast. Example, myoblast, monoblast, which eventually mature into fully differentiated blood cells. Monoblasts develop into monocytes. Eosinophilic myoblasts develop into eosinophils, and other blasts follow a similar pathway towards their respective mature blood cell types. So this is an hematopoietic stem cells which is responsible for formation of an various other types of the blood cells like an erythrocytes, thrombocytes, monocytes, neutrophil, eosinophil, basophil, T lymphocytes, B lymphocytes, and natural killer cells. So myeloid cells are responsible for forming especially RBCs, platelets, as well as hub part of the WBCs, where the lymphoid stem cells are basically forming the lymphocytes, mainly associated with an T lymphocytes, B lymphocytes, and natural killer cells. It's a very easy part to understand about. So this is nothing but an types of the blood, or you can say that it's an form elements of the blood rather than types of the blood, considering that is figure illustrates that how the bone marrow is responsible for forming the different types of the blood cells like an red blood cells, platelets, as well as the white blood cells and its form elements. So blood clotting mechanism is one of the very important blood clotting mechanism involving three pathways: intrinsic pathway, extrinsic pathway, and common pathway. Blood clotting is the process by which blood forms the clot, and according to this pathway, it is a series of chemical reaction that culminates in the formation of fibrin threads. So it is complex cascade of enzymatic reactions in which it factors activates many molecules of the next one in a fixed series. So as the slide indicates, clotting is the process by which blood forms and clot. It's a protective mechanism actually. Okay, so these are the clotting factors. You have to remember these clotting factors as one basic formula: Fipro-T Caladusta anti-ABCD in between Stewart factor at last fibrin stabilizing factor. I am repeating that one: Fipro-T Caladusta anti-ABCD in between Stewart factor at last fibrin stabilizing factor. So fibrinogen, prothrombin, tissue factor, calcium ions, labile factor, stable factor, anti-hemophilic factor A, B, C, D in between Stewart factor, and at last fibrin stabilizing factors. Intrinsic pathway, it leads to the formation of prothrombinase with fewer steps which occur rapidly. Extrinsic pathway, it leads to the formation of prothrombinase with more number of steps which occur more slowly. Common pathway, once prothrombinase is formed, the steps involved in next two steps of clotting are same for both the pathways, called as common pathways. Prothrombinase converts prothrombin into the enzyme thrombin. Thrombin converts soluble fiber content into insoluble fibrin, and fibrin forms the clots. So what it indicates? Intrinsic pathway involves the internal factors, and extrinsic pathway basically involves the external factors, and it is more complex than that of the intrinsic pathway. Common pathway involves basically for the formation of prothrombinase and thereby at last the formation of an clot. Okay, so this is an mechanism of extrinsic pathway and intrinsic pathway. Whenever there is a damage to blood vessels, that is going to activate some clotting factors, like, and as far as concerns about the extrinsic pathway, it activates the factor 4, 5, 7, 10. The common most here in the intrinsic pathway, it involves the additional factor number 11 and 12. So it is responsible for tissue thromboplastin, activation of an tissue thromboplastin, and in intrinsic pathway platelet thromboplastin. So both are responsible for active inactivation of the factor 10, that is going to the active factor 10 with the help of an factor 5. And vitamin K also plays a very major role in the formation of an clotting. So in between these, the common pathway is activated, and it is resulted into prothrombinase enzyme, which is responsible for conversion of prothrombin to thrombin in presence of the calcium ions, and that fibrinogen is also converted to fibrinogen to fibrin, and at last, with the presence of factor 12, it leads to the formation of an blood clot. What is intrinsic pathway? It consists of fewer steps and occurs rapidly within 10 seconds. In this pathway, a tissue protein called tissue factor leaks into the blood from cells outside the blood vessels and initiate the formation of prothrombinase. Tissue factor is a complex mixture of lipoproteins and phospholipids released from the surfaces of damaged cell. In the presence of calcium, tissue factor activates factor 10. Once factor 10 is activated, it combines with factor 5 in the presence of calcium to form the active enzyme prothrombinase, completing the extrinsic pathway. Extrinsic pathway. This pathway is more complex and occurs more slowly, usually in several minutes. The activators are either in direct contact with blood or content within the blood vessels. If endothelial cells are damaged, blood comes in direct contact with collagen fibers in connective tissue around the endothelium of the blood vessels. Injury to endothelial cells, damage to platelets resulting in the release of phospholipids by the platelets. Contact with collagen fibers activates clotting factor 12, which activates clotting factor 10. Platelets, phospholipids and calcium participates in the activation of factor 10. Once factor 10 is activated, it combines with factor 5 to form the active enzyme prothrombinase, completing the extrinsic pathway. Summary of blood clotting. Blood clotting pathways involves extrinsic, intrinsic as well as an common pathway. So finally we can conclude that the conversion point is for both the pathways is at the formation of prothrombinase, which converts prothrombin to thrombin, and finally thrombin converts fibrinogen into fibrin, resulting in a blood clot. So what is common pathway? Formation of prothrombinase is the starting point of an common pathway. In the second stage of blood clotting, prothrombin and calcium catalyze the conversion of prothrombin to thrombin. In third stage, thrombin in presence of calcium converts fibrinogen to loose fibrin threads. Thrombin also activates factor 13, that is fibrin stabilizing factor, which strengthens and stabilizes fibrin threads into a sturdy cloth.